thank all of you for making it out in the middle of a busy day. We appreciate the effort because I know it is a very busy day this time. Also, congratulations to those of you with the I voted stickers. I don't have one on, but at least some of you did. So I appreciate that. So we're very happy to have the third of our three IPIT awards. And as many of you know, back in 2007, the Institute for Pharmacogenomics and Individualized Therapy was started. People from around the School of Pharmacy, School of Medicine, School of Public Health, School of Nursing, School of Dentistry, the College of Arts and Sciences have been very active in trying to find ways to individualize therapy. And we quickly realized that there was no way of thanking those who have made our life easier. Many of us have been helped by those previous to us, and we might have even thanked them personally. But there was no national or international award that really brought together those who have helped us with personalized medicine. And using the term broadly, those who have done things to help with patients, with the public health, and with the clinical aspects of individualized therapy. And so we started these awards. And this is our fourth year of giving these awards out. And we're very pleased to today give the 2010 award for the IPIT Award for Clinical Service. Now this is an award we give out annually for people who have made significant direct contributions to the advancement of individualized therapy in clinical practice. And so we gave an award out a few weeks ago to Moon Curry for his work that involved public health and the broader aspects of pushing individualized therapy. But today we're focused on ways of trying to help individual people in this field. And we're very pleased that the awardee for 2010 is Dr. Mary Relling. Now, Dr. Relling is well known to many of you here through her interactions with pharmacy, with pediatric hemonc, her activity in all aspects of clinical pharmacology. She's the chair of the Department of Pharmaceutical Sciences at St. Jude Children's Research Hospital and is a professor of both pharmacy and medicine at the University of Tennessee in Memphis. She received her PharmD from the University of Utah, where she also completed her residency training, and I will omit the year in which that happened.
Thank you, Howard.
She completed her fellowship training at St. Jude's, where she has been on faculty since 1988, and has grown through the ranks there at that institution. She's published over 200 original scientific manuscripts in prestigious journals such as JAMA, New England Journal of Medicine, Science, Lancet, etc. Is a frequent national and international guest lecturer, and has served as the chair of national committees in all of the prestigious oncology and clinical pharmacology organizations. She was also elected to the Institute of Medicine in 2009. Now, Dr. Relling was selected by a committee of members of IPIT, and a committee that was chaired by Dr. Richard Goldberg, they call it here, who is the physician chief of the cancer hospital and the chief of hemonc here at UNC. And the committee looked at a number of different individuals and chose Dr. Relling because of the dramatic and revolutionary improvements she has made has caused in the area of individualized therapy. And we like that word caused because much of the work she has done is not necessarily improving on or following existing work, but rather causing elements of the field to be created, opening up aspects of individualized therapy that just never had been thought of before, or for which there's only theory and no data. And so this idea of really pushing this forward has come from understanding mechanism in real people, rather than just cells, all the way through to some of the work she'll talk about where they're driving the choice of therapy for both cancer and supportive care using genetic information. So Mary, if you could come up here. Second, Rich, if you could come over as well. Because of my white shirt, we're kind of going to take it from this angle. But I'd like to present to you the IPIT Clinical Service Award. So,
I was going to do that for him. Thank you.
Yeah,
I'm glad you did.
Is my collar okay? There you are, Mary.
Thank you so much. Thank you. Great. Thank you. That's beautiful. Thank you very much. Oh, thank you. Thanks. Thanks, Howard. This is a total pleasure to be here, and it's an honor to receive this award, which is really the thing that I care about most, which is trying to figure out how to do important research, but also how to actually incorporate what we can into clinical practice now. Also, when you were talking about thinking of things people haven't thought of before, I don't think I've ever thought of anything that nobody has thought of before. I think I'm a big believer in persistence and stick-to-itiveness, and that's part of what staying at the same place for more than 20 years allows you to do. So I will, shall I use the mouse for a pointer? Yeah, that's probably the best. So I'm going to talk about acute lymphoblastic leukemia. If you want to,
I take it all back.
You want to talk about a leader. This was taken shortly after 9/11 when we were trying to get from a hotel onto the NIH campus, and there was so much confusion about security that the bus driver wasn't going to let us through. So Howard jumped into the driver's seat and commandeered the bus and got us onto the NIH campus, I think. And we decided to capture that in film. So it's really Howard, as you all know, from his incredible charisma that has moved the field forward probably more than anybody else. And it means a lot to get this award from you and your institute. So I'm going to talk about pharmacogenomics, but if we first want to think what's the importance of inherited genomic variability, I think there's a couple of problems still facing us. One is that it is still unclear whether common versus rare variants are going to be important. And as I was just talking about to the students, whether genomic variability is going to be important but so rare that it may be untenable to find and act upon. And we're all trying to find those things that are common enough and findable enough with high enough odds ratios that we can discover them and act upon them. And then do we think that the phenotypes we're interested in, like drug response phenotypes or cancer risk, are likely to be inherited? And I'd like to go back to these data from twin studies, a large study of monozygotic versus dizygotic twins of 40,000 twin pairs in the Nordic countries, and looking at some common adult cancers, colon cancer, breast cancer, prostate cancer. It's estimated that around 30 to 40% of the risk of developing this complex phenotypes of adult cancers are inherited or are heritable phenotypes. Likewise, when we're looking at pharmacogenetic phenotypes, we don't have the luxury of giving anti-cancer drugs to pairs of monozygotic versus dizygotic twins, but other drugs have been administered to twin pairs. And by looking at the variability among versus between those different kinds of twin pairs, it's been estimated that for a test substrate like antipyrine, 75 to 85% of the pharmacokinetic variability among individuals could be due to genetic inherited variability. Now, we can't give anti-cancer drugs to normal volunteers, but through work that Howard pioneered in this publication several years ago, along with Eileen Dolan's group, where lymphoblastoid cell lines are treated in vitro and survival is measured versus drug concentrations, and the familial relationships in these extended kindreds can be analyzed. One can estimate the heritability of the susceptibility of cells to undergo apoptosis as a result of exposure to anti-cancer drugs. And putting all those data together, probably something like 30 to 70% of pharmacodynamic variability can be attributed to heritability or to genetics. But still, even though we can probably estimate that a high percentage, 30 to 90% of variability among individuals is genetic, how much of it is attributable to a tractable number of polymorphisms? And that's kind of where our research and many other people's research is focused right now. Now, in our own work, I focus on childhood ALL. Acute lymphoblastic leukemia is the most common childhood cancer. And the outcomes in this disease have risen dramatically from the 1960s to the current status where it's 85 to 90% event-free survival. However, some patients are still at unacceptably high risk of relapse, and some survive but with unacceptable toxicity such as second cancers or other severe debilitating long-term effects. And so our research is focused on estimating the extent to which pharmacogenetic variability contributes to the variability in these important phenotypes in childhood ALL. I think ALL is a great model for pharmacogenetics. It is the most common childhood malignancy. It's a life-threatening disease that's cured with dangerous medications. And there's a culture of using genomics to adjust the intensity of therapy. Now, albeit those genomics have generally been the genomics, the somatically acquired genomic variations of the tumor cells, but that's built into the way in which clinicians treat this disease. And importantly, about 85% of patients in the U.S. with this disease are treated on clinical trials through Children's Oncology Group, St. Jude, or Dana-Farber trials. Non-genetic prognostic features and covariates are very well defined and are captured as part of these clinical trials. And there's high-quality germline and tumor DNA prospectively collected in the Children's Oncology Group since at least 2003. And in, at St. Jude, actually going back to probably about the time when Howard was a fellow in the early 1990s. So this gives us the opportunity to go back and do a lot of genomic studies. And many of the medications that we use in ALL have crossover application in other adult malignant and non-malignant settings. So I'm just going to review a couple of challenges and findings. And one of the first points I'd like to illustrate is that the relationship that we find between genotype and phenotype will vary with treatment protocols, especially after we start using genomics to alter the therapy that we use. And as an example of this, if we look, I know you can't read all of the rows on this slide, but this is a typical publication from St. Jude or any other ALL group where one of the tables includes all the possible prognostic features of these patients, risk group, age, race, CNS status, et cetera. And then, was this prognostic feature actually prognostic in this particular treatment protocol? Is there a significant p-value relating these patient characteristics with the phenotype of interest, that is event-free survival? And actually, many of the traditional prognostic features that we use in ALL weren't present in this trial, trial total 13B at St. Jude, because it was very aggressive therapy that abrogated and basically wiped out the importance of lots of, quote, traditional prognostic features, including the presence of the TEL-AML1 translocation in the leukemic cells themselves, where this was not prognostic at all, a p-value of 0.41 in this particular trial. Now, that doesn't mean that TEL-AML1 doesn't have importance for the biology of ALL, and so several groups, including ours, showed many years ago that the acquired genomic variation of the presence of the TEL-AML1 fusion was strongly associated with relapse with those individuals who had the translocation having a better outcome than those that did not in trials conducted in the 90s. But again, this prognostic significance completely disappeared in later trials that probably included more aggressive therapy. So another example of that will be provided by the pharmacogenomic polymorphism in thiopurine methyltransferase. This is one that Howard worked on with Dr. Evans. It's been known for some time that there's a polymorphism in the TPMT gene. It codes for an enzyme activity that results in a polymorphic distribution, a trimodal distribution of the population, with a small percentage being homozygous for variant alleles, 10% being heterozygotes, and 90% of the population being wild type. It shunts active drug or prodrug away to an inactive metabolite. It prevents its anabolism to the active thioguanine nucleotide metabolites. And so this translates into very low concentrations of the TGN metabolites in those who are wild type and very high concentrations in those who are homozygous for the variant allele. And this is just illustrated here, that there's much higher levels of the active thioguanine nucleotides if you're given a standard dose of 6-mercaptopurine, if you're homozygous deficient versus carrying one variant allele versus homozygous for the wild type allele. And if you give patients this standard dose of 6-mercaptopurine, that translates into a relationship between toxicity and the TPMT genotype. So the proportion of patients who require a dose decrease to avoid myelosuppression is all of those who are homozygous for the variant allele, about 35% of those that are heterozygote, and only about 7 or 8% of those who are wild type. This was recapitulated in a murine model of the thiopurine methyltransferase polymorphism. And so the mice that could avoid life-threatening toxicity of 6MP were all of the wild-type mice, a proportion of the heterozygotes, and all of the homozygous knockout mice succumbed to toxicity given a fixed dose of 6-mercaptopurine. And it really recapitulated the findings that we saw in patients very well. So before we adjusted doses of thiopurines based on TPMT genotype, there was a trend for better event-free survival, leukemia-free survival in those who had at least one defect, because they have higher concentrations of the active thioguanine nucleotides than those individuals who are wild type. But starting probably about the time that Howard was a fellow at St. Jude, I don't know, is that about right? We started modifying the dose of 6-mercaptopurine prospectively based on TPMT status in the patient, so that those who are wild type got the full dose, heterozygotes got a reduced dose, and homozygous variant individuals got a vastly reduced dose of 6-mercaptopurine to avoid acute toxicity and myelosuppression from those high active metabolite concentrations. Now, when we go back in this study to look and see is there a difference in the cumulative incidence of adverse effects of dose-limiting myelosuppression based on TPMT status, there's no statistically significant difference at all between those that have a variant of TPMT and those that are wild type. And that makes sense because we use that feature to adjust the doses of thiopurines in these patients. And most importantly, there was also no difference in the risk of relapse in the patients who had at least one variant copy of the gene versus those that were homozygous wild type. In fact, if anything, it trended to be a lower risk of relapse, even though the heterozygotes got a lower dose of the 6-mercaptopurine. And so there's a couple of implications of this. One is, of course, that if we had been trying to replicate the finding associating TPMT genotype with either relapse rate or acute toxicity, we, of course, wouldn't find it in a study in which TPMT itself was used to adjust the doses of thiopurines. And so that genotype-phenotype association doesn't have a chance of replicating in a trial like this. And also, of course, we think that this means that it's important to try to use TPMT status to adjust the doses of thiopurines in ALL therapy to avoid acute toxicity without compromising the cure rates. Of course, we want to discover other variants that are associated with relapse, and so we've been using genome-wide tools to assess what some of those other genes might be. And now when we're talking about responsiveness, we assume that both variation in the host genome and the acquired variations that are present in the tumor cells themselves are likely to affect relapse risk, where, of course, host variations are more likely to affect host toxicity. So one of our first questions before embarking on these genome-wide studies was to understand whether there could be some confounding. Could germline polymorphisms differ depending on the ALL subtype? In other words, could they differ among ALL subtypes that had different acquired genomic variation? Might there be a spurious association between germline genotypes and responsiveness that was actually due to an underlying confounding of germline variation with the type of leukemia that the patients developed? And these ALL subtypes with their acquired genomic abnormalities are the presence of the E2A PBX fusion or TEL-AML1 or the Philadelphia chromosome positiveness can have a substantial effect on the prognosis of this disease and have very different clinical outcomes. And we're interested if we can define what the inherited variations are that might affect these different outcomes, and also what the mechanism is by which these acquired variations affect drug responsiveness. Now, from Bill's work, we've known that different ALL subtypes have very different accumulation of drugs, including, for example, methotrexate polyglutamates. So this is the methotrexate polyglutamate accumulation in B hyperdiploid ALL versus those carrying the E2A PBX, T-cell ALL, and so on. And so we knew that, for example, subtype of leukemia could have a big effect on this drug accumulation phenotype of methotrexate polyglutamates. So before we look for germline variants associated with methotrexate polyglutamate accumulation, we want to understand this potential confounding and look for germline genetic variations whose allele frequencies could significantly differ among ALL subtypes. And we did this using a genome-wide approach. We used children that were genomically defined as European whites only, because we had to define the control group, which was whites only. We had children with ALL from St. Jude and COG studies, and our controls were from the Wellcome Trust and the GAIN bipolar and schizophrenia group subsets that are publicly available. We typed the germline DNA from our patients with the Affymetrix 500K array. And then we compared allele frequencies in our discovery cohort of children with ALL compared to this combined non-ALL control group of almost 18,000 individuals to identify SNPs that were significantly different between children with ALL and large group of adults without ALL. And then from those SNPs that distinguished ALL from non-ALL, we further distinguished SNPs that might differ in frequency between subtypes, so between B hyperdiploid and all other subtypes, between TEL-AML1 and all other subtypes, and so on. And looking at the Manhattan plot for this genome-wide association study, the inverse p-value versus the chromosomal location, there were a couple of polymorphisms that were highly significantly different between the ALL cases and the combined non-ALL control groups. Two of them were in the ARID5B gene, and one was in this Icaros gene. So there were 6 genotypes that distinguished ALL subtypes, not just ALL from non-ALL, but from the subtypes. And 2 of these replicated at the p less than 0.05 value in an independent cohort of individuals. So the ARID5B SNPs did distinguish hyperdiploid B lineage ALL from other types, and the Icaros subtype had a p-value of right at 0.05 in the replication cohort that distinguished T-cell ALL from all other subtypes of ALL. And interestingly, at the exact same time, an independent group working in the UK compared children with ALL to controls from the UK 1958 birth cohort. And another group of controls, they used a completely different platform, the Illumina 550K instead of the Affymetrix 500K. And they found exactly the same 2 top hits that we did. So in bright blue are the SNPs that were found in the US cohort, and you can see the odds ratio of developing ALL versus control was between 1.6 and 1.9 in distinguished hyperdiploid, and it was an odds ratio of 1.7 in the British group, and also distinguished the hyperdiploid subtype of leukemia. For Icaros, we had an odds ratio of about 1.7, and it was exactly to 2 decimal points the same odds ratio for a SNP that was in LD with our original Icaros SNP, again, on this independent platform. These are plausible candidates. ARID5B is a transcription factor that's widely expressed. The knockout mice display aberrations of T and B cell lymphoids, T and B lymphoid cells, and it's involved in lymphoid differentiation. Icaros is also critical for lymphoid development, and its deletion has recently been shown to be strongly associated with poor prognosis in ALL. And if we go back to our original, one of our original questions, that hyperdiploid ALL had the highest intracellular methotrexate accumulation of any subtype. And And when we look at the accumulation of methotrexate relative to the ARID5B SNP, we see that the polymorphism also affects the methotrexate polyglutamate accumulation. So we have an inherited variation that affects the subtype of leukemia that the patient is going to develop and penetrates through and affects how that subtype accumulates drug in a way that is consistent with its good prognosis and its good responsiveness to anti-metabolite therapy. So we think the implications are that it is possible that genotypes are associated with drug response, are also associated with ALL risk and ALL subtype risk, and that we have to account for subtype, or at least explain for confounding effects of germline genotypes with drug response phenotypes. And we've also shown more recently that ancestral differences in the frequency of these alleles can explain race-related differences in the frequency of this disease. So we know that the incidence of ALL in whites is about twice what it is in blacks in the United States. And Wenjin Yang and our group went on to show that you could account for about 30% of the higher risk of ALL in whites simply by the difference in the prevalence of the risk allele in ARID5B in whites compared to blacks. And that's illustrated based on the hatched areas here in the whites compared to the blacks for the 4 different major subtypes of leukemia. And in fact, it looks like this single polymorphism can account for several of the race-related differences in the frequency of this disease. So we also have looked at how genomic variability using a genome-wide approach can contribute to early treatment response, minimal residual disease. And this was work led by Jun Yang, a young faculty member in our group. Again, a collaboration between St. Jude and the Children's Oncology Group. And we looked for those polymorphisms that were in common, that were associating with the presence of minimal residual disease in 2 unrelated cohorts that received somewhat different therapy, were actually assessed at somewhat different times for their MRD responsiveness. And at this p-value threshold that was chosen to minimize false discovery rates, there were 102 overlapping SNP genotypes. As an example, 5 out of those 102 were all in the same gene, the IL-15 gene, and the germline inherited genotype TT, TC, or CC was associated with MRD responsiveness in both the St. Jude cohort in black bars and the COG cohort in white bars. And IL-15 is an interesting gene candidate, again, not one that would have been at the top of our list, but about simultaneously a paper came out from the BFM showing that IL-15 expression in leukemic cells was related to the risk of relapse in this disease. And several studies have shown that IL-15 is involved in susceptibility of lymphoid cells to glucocorticoid-induced apoptosis. So a reasonable candidate, but one we probably wouldn't have discovered without a genome-wide approach. We went on to say out of those 102 SNPs that were associated with clearance of the leukemic cells at the end of induction, how many of them are associated with host phenotypes such as anti-cancer drug pharmacokinetics? We only had a couple of such phenotypes measured in this group: methotrexate clearance, the blast accumulation of the methotrexate metabolites, and etoposide clearance, which is sort of a measure of a CYP3A P-glycoprotein type drug. And 21 out of the 102 SNPs were associated with just host drug pharmacokinetics, 20 out of the 21 in the plausible direction. That is, high drug clearance corresponded to lower drug levels and therefore corresponded to higher levels of MRD. And the inverse was obviously also true. So of these SNPs that were associated with the response, only about 40% of them were within genes, as has been seen in many genome-wide studies. And we think it's a combination of ALL-related polymorphisms, like the IL-15, which is probably just shining through and having its penetrative effect in the leukemic cells. And some of them are probably host-related, and a surprisingly high proportion seem to be related to host pharmacokinetic variability. That is, ability to clear MRD may just be partly dependent on how much drug is circulating. So I want to close with a few minutes more about clinical translation, because I'm confident that the work that we're doing and Howard's group is doing and other groups are doing are going to continue to find important genomic variation that is associated with drug response and with toxicity, and some of those things will be worthy of incorporation into clinical practice in the future. But there's a lot of personalized medicine that we could do right now, but generally it's not being done. We do have a little bit of an exception at St. Jude. As I'll show you, we've been genotyping for TPMT and the CYP2D6 gene in our ALL patients for a couple of years. So I think there's a lot of barriers to integration of pharmacogenetic tests into clinical care. One is the fragmentation of healthcare systems, especially over a lifetime, right? We're going to be generating a lifetime test, a genomic test, that should stay with the patient forever, but we don't have anything close to a universal medical record that stays with the patient forever. So how is this information actually going to stay with the patient from healthcare system to healthcare system to healthcare system? Our healthcare system and most payment structures are focused on sick care and not on disease prevention. In some cases, a lack of evidence on clinical utility or cost effectiveness is slowing pharmacogenetics down, but as we were talking about earlier this morning, it's coupled with excessively high rigor to which genomic tests are being held compared to many other tests that are routinely incorporated into the clinic, and that even has its own special term of genetic exceptionalism. There is a challenge underlying interpretation of the laboratory results, and there's a real lack of computational decision support in all of medicine. It's still a remarkably small percentage of any healthcare systems that are close to 100% EMR, electronic medical record. And this includes the medication process, which will go all the way from testing, prescribing, distribution, and administration of medications. And another barrier is, I think, the need for preemptive testing. And I know Howard has written about this extensively, that to be practical, these tests need to be available a priori in the background as part of the patient characteristics, and testing at point of care as the idea enters into the clinician's head is probably not going to be feasible in a temporal fashion or in a financial fashion. And we think that we can either overcome or ignore essentially all of these barriers at St. Jude. With the exception, of course, that we can't force the patient medical record to follow the patient for a very long time, but because our children generally are diagnosed as children, they stay in our care, at least as related to their cancer care, for essentially the rest of their life. We have a long time to provide this information for them. So we're starting a clinical protocol that's called PGen for Kids, and it's to do clinical implementation of pharmacogenetics on a broad scale. Our goal is to migrate pharmacogenetic tests from the laboratory across that firewall into routine patient care so that the tests are available preemptively. It involves a lot of investigators at St. Jude, including lots of pharmacists, our information sciences, our chief medical information officer, social workers, research nurses, the heads of all of our clinical services, statisticians, our representative on our patient advisory council, and many others. And we're collaborating with Uli Brockel at Medical College of Wisconsin for genotyping. So the process looks kind of like this. The patient is enrolled. Their DNA is genotyped using a high-throughput platform, such as the DMET array, with all of the results being generated in a CLIA-approved laboratory. So assuming the basic quality control is met, the genotypes will be classified as clinically eligible genotypes, or for some other reason, suspect and not allowed in to be eligible to go to the medical record. And the vast majority of all of those genotypes will stay over in the laboratory research firewall, stay in the research databases. But based on evaluation of how pharmacogenomic data are progressing and the association between genotypes and drug phenotypes, we will use an approach to identify a small subset of those clinically eligible genotypes that meet the threshold for a clinical pharmacogenetic locus and go over into the medical record. Those genotypes will be posted into the electronic medical record with a basic pharmacogenetic consult provided by one of our pharmacists, and a very small subset of those results will be considered to be high-risk genotypes that should not only go in the medical record, but should be supported with electronic decision support to have a warning fire if an attempt is made to prescribe one of the affected drugs. Sorry about all the animation. So which drugs do we think are the highest priority? And these are the same drugs that I'm sure you have on your list here. TPMT and thiopurines. We think CYP2D6 and codeine is one that should go into our EMR quickly. Almost all of our kids with ALL have incretin neuropathy, and they're usually prescribed codeine. What other drugs should go in is a source of a lot of controversy. G6PD and rasburicase, whether we should include Septra, is a big controversy. UGT1A1 irinotecan, the warfarin genes, 2C19, with clopidogrel, which we hardly ever use, and maybe something like voriconazole, which we use a lot, DPD and 5FU, and then, of course, all these HLA reactions, genotypes that are frequently tested for now. Most of these tests are included on the Affymetrix DMET array, and as is well known to you, a lot of the important ones are not. And even some of those that are incorporated into the DMET array are probably not usable in their current format. This set of gene-drug pairs is highly compatible with the list of gene-drug pairs that we're working on in a national group called CPIC, Clinical Pharmacogenetics Implementation Consortium, where we've surveyed individuals in pharmacogenetics and clinical pharmacology and prioritized many of these gene-drug pairs. Of course, conducting a survey of experts is not necessarily a good way to get a good list. I figured that out. You have to just say, well, they didn't know what they were talking about. So this group is using PharmGKB to store its information. We have groups that represent scientists, FDA, third-party payers, et cetera, and we're hoping to provide updated recommendations on exactly how to use genotype results to do drug dosing in the future. So one of the things we've talked about is what are our criteria for prioritizing gene-drug pairs. Again, I think these are similar to what you guys will use and are using. Impact of professional organization recommendations can be helpful. FDA labeling can be helpful. Evidence that third-party payers are already reimbursing for these gene tests one at a time can be helpful. Lawsuits who've penalized clinicians who haven't used the gene test can be helpful. The availability of standalone CLIA-approved tests for those individual loci. And then, of course, the data that go into all of those more tertiary things, such as clinical trials that demonstrate the drug effects are linked to the loci, a narrow therapeutic index for the drug, preclinical studies that demonstrate the functional basis of those associations, and in vitro or in vivo evidence suggesting that drug A responds just like drug B. So those are all pieces of evidence that will be used by us and by other groups in helping to prioritize what makes something be so compelling that it should move into the medical record and that one should act on the information if one has it. And so then those clinical pharmacogenetic loci will move over into the medical record. And once we make the decision that warfarin 2C9 VCOR-C1 should go into the medical record, it will go into the medical record for all patients previously studied and all patients to be studied in the future at that time. Because an important concept of this is having it be preemptive. It's not the prescribing of the drug that is the thing that moves the genetic test across the firewall from the research lab into the medical record. It's the fact that any evidence strongly links that gene variation to prescribing that drug, whether or not that patient is likely to be prescribed that drug. So this is the way it looks in our EMR. The pharmacogenetic test, this is the thiopurine S-methyltransferase genotype result, gets entered in with the designation here, star 1, star 3A. Of course, that doesn't mean much to most clinicians, but that's the way the laboratory test would be entered in. And if you click on this result itself, you'll get to a consult that will have more information about what that patient's genotype is, what that means, and what the likely implication is for drug dosing in that patient. For that small subset of genotypes that are high risk and should be supported by decision support firings of warning flags, and we count on our colleagues in information sciences to help us get these built into our EMR. So I want to close with that all of the work that we're doing in the research side of things and in the clinical implementation side involves a lot of talented people in our department, including many postdoctoral fellows, graduate students, and pharmacists that work with our patients day after day. Our Hartwell Center for Bioinformatics and Molecular Technology, statisticians, other clinical collaborators at St. Jude, my collaborators in the Pharmacogenomics Research Network, and importantly, our collaborators at the COG who've really shown tremendous forethought in making sure that we get genomic material for all patients enrolled on these studies. We enroll almost 3,000 children a year with ALL on our frontline COG study, so this is going to be a tremendous resource. So I want to thank you again for your attention. Thank you again for this award, and I'll be happy to take any questions.
Time for some questions for Dr. Relling. Maybe I could start off, Mary. One of the things you showed early on were the slide of the great improvement in outcome from childhood ALL. And we've talked before about how that was done with virtually no new drugs. But it was done with a lot of new molecular markers. And childhood malignancies were very aggressive at putting molecular markers in. And aggressive about understanding old drugs. So I wonder if you could maybe give us some advice from that on, how we should be doing more of that. Because as you know, there's this huge divide between success on the adult side and success on the kid side.
Right.
In terms of outcomes.
Well, the number one step is to enroll people on clinical trials. And we've enrolled patients on clinical trials even when they've been extremely boring phase 3 trials where there's almost no interest in the randomized question, but we've gotten good biology in every single person who enrolls on the trial. We know what therapy they got, and we know what toxicities they had. And really for all of individualized medicine research and pharmacogenomic research, we don't need the phase 3 randomized therapy questions to even be present. We just need the phase 3 therapy to be present and to know what it is. So I think that the key is to enroll patients on clinical trials. And I'm sure you're right that the combination of using good molecular markers, learning from past trials to slightly change the therapy so that essentially every generation of studies has resulted in a 5 or 10% increase in event-free survival from the previous generation. And if you do the math, you go from the 1960s to the 1990s from 10% to 90% just by making those incremental increases. So I know that not all adult tumors are going to be as chemo-responsive as childhood ALL is, but I don't see how you can make the progress without being willing to enroll patients on phase 3 studies. And that's been a huge accomplishment of pediatric oncology in general, and St. Jude in particular, but really the cooperative group does just as well. I don't know, what do you think?
Well, I think that's a major part. If I knew the answer, we'd be doing it.
Yeah.
Bill?
Hi, Mary. Could you talk about the potential disconnect between genotype and phenotype, and is it ever going to be practical to test for phenotype instead of genotype because of all the logistical issues?
Well, it depends on the gene, right? So my understanding is for DPD, you might still rather do DPD activity than try to impute DPD phenotype based on genotype, because there's such a preponderance of rare variants, you'd be scared to say that you ever got somebody's genotype correct. Whereas luckily, 20 years of research has shown that for TPMT, with 3 polymorphisms, you will capture well over 98% of all the inactivating variants. So you'd be much more willing to call your phenotype based on genotype. I mean, I love phenotype if phenotype is stable enough and inexpensive enough and you can get the right tissue so you can measure it. There's nothing wrong with phenotype. Also, TPMT, we have that luxury that there are actually, CLIA-approved commercial tests that allow you to measure TPMT activity phenotype, thiopurine metabolite level phenotypes, as well as TPMT genotype. And I do get phone calls and emails from all over the country of cases where people are confused. And sometimes maybe there was a sample mix-up, because sometimes you get a different answer from genotype than phenotype. That's something that scares us a lot about genotyping, is sample mix-ups. Because the problem with a lifetime result is you better have gotten the right sample from the right patient. And we see mix-ups more than anybody would like to acknowledge. So we're putting in secondary samples for every single patient to do a secondary method of sample identification, because I'm scared about getting the wrong answer into somebody's chart.
Dan?
Yeah. Early on in the talk, you had an example, I believe it was TPMT, where you had a group of patients who had the most toxicity but also had the best efficacy. Can you just talk through the discussion around how you decided what to do with the doses in the homozygotes, heterozygotes, and how you decided whether you should decrease those doses or increase other doses to bring the other patients up to that level, and where you think the right risk-benefit optimizing point?
Yeah, I mean, again, luckily we weren't starting from scratch. There were 30 years of history of using 6-mercaptopurine in ALL, so we kind of knew what the maximum tolerated dose was in the population. And you could assume that that was being driven by the 90% of the patients that were wild type. So we knew pretty much what the wild type patients should be getting. The other good news is it's a drug that's given orally, daily, for 2.5 years, monitored by weekly white blood cell counts. And so there's generally, it's not just genotype that we use to decide on the dose. That's what we use for the starting dose. But we can titrate the dose subsequently based on a desired level of neutropenia. The one exception that we have for TPMT, for those who have variant alleles, is we are a little bit scared about using myelosuppression as the titration endpoint, because there's an association now between TPMT status, thiopurine use, and long-term secondary cancers that our group found and several other groups around the world have found. So that makes us a little scared to go up on the dose of thiopurines to the normal level in the heterozygotes, because we know that they had been at higher risk of secondary tumors. So that we actually have a cap on the dose that we chose somewhat arbitrarily based on what their metabolite levels are, what their increased risk of second cancer is. We may not be exactly right on where that cap is, but we had a pretty good idea of where to guess that it should be. But again, that was a luxury to have, which we may not have for all drugs, so much information about how to try to dose it right. Pharmacogenetics can help.
Can you
tell me which genotyping platform do you use for those kits for 26 genotyping, and who is paying for that?
So for the 2D6 genotyping, we've been sending the samples to ARUP, and they do some allele-specific test. I don't know exactly what it is. It is a real question for us now because we have discordant results in our genotyping for 2D6 by the clinical lab that's already in the medical record, and the genotyping results by the DMET array. which is insensitive to copy number variations, apparently. So now we're going to be forced to use a third method to really confirm that those cases of ultra-rapids and deletions are what we think they are, but I guess I'm leaning towards the ARAP method being correct. Who's paying for it? For TPMT, we are doing third-party billing for that. For CYP2D6, I am not positive if we're doing third-party billing or if we're just eating the cost of that. It is part of a research protocol. That doesn't mean it's not a billable test. We have all of our ALL therapy as, quote, part of a research protocol, and of course a lot of it can be billed to third-party payers because you would do the testing and you would do the drug prescribing anyway. CYP2D6 you could argue about a bit, but I would be willing to try to get it. I would be willing to try to get it paid by third-party payers because we have data that something like 80 or 90% of the patients would be prescribed codeine. So to test all of them to find the 15 or 20% that are going to have problems, I think you could justify, but you may know better than I do.
So seems there are still about 10 to 20% of patients can be cured by the existing treatment. So is there any genotype difference between this group of people with the rest of the 80%?
Yeah, well, we're looking. Yes, there are some. Whether they are universal or whether they're only in some subsets of patients, we're still trying to figure out. And that's going to be a real challenge if you're only trying to identify the genotypes that set aside the 10% who fail. If it's not the same 10% who are failing in treatment protocol 1 and treatment protocol 2 and treatment protocol 3 and treatment protocol 4, you're going to have a real hard time replicating the genes that may be putting some patients at risk. So far we only know it from studying a few thousand patients, and I think we're going to need to study several thousand more before we're comfortable saying what those genes are. And some biology, obviously, to back up how they might do it.
Dan,
For your PGen for Kids initiative, are you collecting any data right now on how many codeine reactions you're having before implementation of this to compare how effective the...
No. The only gene tests that are going to move over into the EMR are the ones that we think all the data are there. I mean, of course, it's always nice to have more data, but we're not going to generate more data showing that that genotype is important for that drug. The drug is going to have proven that it's important for that gene by other people's or our own earlier research. It's just the only reason to have a protocol instead of just doing it clinically is the fact that we think we have to have consent for withholding results. So if we do a DMET array and we generate 1,900 genotype results, but we're only willing to put a small fraction of them into the EMR, we think we have to get consent from the patient to say we're not going to put some results in the medical record. And we may be wrong about that. Like at Vanderbilt, Dan Roden, their consent up front to be treated at Vanderbilt says, I consent to you using genomics to treat me, and you have to opt out of that if you don't want that done. And they think implied in that consent is that there may be a lot of genetic testing that's done that doesn't ever go into the electronic medical record. And I can see making either argument, but right now we're starting, partly because they're children, we're starting a little more conservatively.
Well, with that, we'll thank you very, very much for